Okay, class, so we are done with the laboratory procedures. So let us proceed sa aton nga, although this is 4.2, 4.3 ang nabutang sa babaw. So we are now on your agar and broth. So what are your agars and broth? So it is very important sa aton nga microbiology, kapin na sa aton bacteriology, ang agar kag broth because that is the ginagamit ta pinapatubuan sang aton nga mga microorganisms. So, a quick introduction sa aton nga agar. So, it is with its distinctive smell. Okay, one can easily distinguish agar from other materials commonly found in your laboratory. Chemically, your agar is a polymer made up of subunit of sugar galactose and its component of the cell wall for several nga species of your red galaxy and usually harvested in the Eastern Asia and California. Okay, dissolving in a boiling water and also cooled, your laboratory agars looks gelatinous or daw gel-like. Although your agars chef use in your culture medium for various microorganisms, particularly for bacteria. Its others well-known use include the serving as that thickening paraton nga soup and so mga sauces in jellies and also ice cream kag sa cosmetics for clarifying beverages and also sizing your fabrics. Now, one might ask why agar, as opposed to regular gelatin like found in your jello, is used for culturing your bacteria. Okay, the answer is agar. Unlike your gelatin, okay, would be degraded or eaten by your bacteria. Okay, kay kung may magtubo sa nga bacteria sa aton nga gelatin, okay, gina-consume niya ang gelatin. Compared mo sa agar, if may magtubo da nga bacteria, okay, only the glucose or the sugar component sa aton nga agar ang i-consume sang aton nga mga microorganism. Okay, wala ka sa may makita gid nga mga breakages gid. Okay? Then also agar is firmer and also stronger than your gelatin. So it is possible, however, to use your gelatin as a culture media for bacteria if agar is not available. So, our laboratory manuals give us information of the uses and design for your agars. Agar is a polycocoloid extracted from a group of your red-purple marine algae, ang tunang baklina, or class Rhopodophyceae, including your Gelidium, Pricklecalydia, and Gracilaria. Okay, your Gelidium is preferred source for your agar because the impurities, debris, and also minerals kag pigments are reduced to a specified level during a manufacture. Agar is a gel at a room temperature and remain firmer at the temperature and remaining be firm at the temperature as high as 65 degrees Celsius. And agar melts at approximately around 85 degrees Celsius and differentiate a different temperature from at which it solidifies around 32 to 40 degrees Celsius. This property is known as your hysteresis. A agar is generally resistant to shear force. However, different agar may be have different gel strength or degree of your stiffness. Agar is typically used in a final concentration of 1 to 2% for solidifying culture media. Okay, and smaller quantities around 0.05 to 0.5% are used in media for motility studies. And you have your 0.5 weight per volume. And for your growth of anaerobes, it is 0.1% and your microaerophiles. A specification for your bacteriological grade agar includes good clarity, okay, controlled gelatination, and your temperature, and your controlled melting temperature. Also, good diffusion characteristic, and absence for your toxic bacterial inhibitors. Okay, so what is the ano yung mga toxic bacterial inhibitors? So the main purpose of your agar is to encourage the growth of your bacteria. Therefore, wala dapat da sang toxic para sa microorganism kaya kundi mapatay ang mga microorganism da. And there is an relative absence for metabolically useful minerals and compounds. Now, the best agar for your student projects. So at least dire will be given ang mga common nga agar which will be pwede nyo ma-handle. Okay, also with the supervision, okay, isang mga teachers or laboratory technician. So, and ano talang pilion talang mga common git. So you have here your common types of your agar. And then let's go. Okay, for your student growing bacteria at home, okay, doon hindi ka rin man kapag growing at home because you also, aside sa need mo mga incubator, para mag-lessen ang contamination sa mga agar bala class, and also oven, sa diin it will give a maintained nga temperature for your microorganism. So, usually ganyan dapat sa laboratory na. Okay? With supervision and also teacher, for example, for investigating a bacterial growth on various places around the house, it is important to use agar formation that does not preferably grow one kind of bacteria over another. The worst case, okay, yung tabo, for example, the worst case would be a preferential grow of your pathogenic nga bacteria kung mapag-grow ka sa imong agar sa imong balay. Diba? Kung hindi mo pag hindi ma-dispose properly, pwede ka mo magkasakit sa imong family. So, therefore, we recommend a plain nutrient agar lang as kung ubrahon yung man sa inyong panimalay. Okay? The common types of your agars are here down below. So, different types of your agar are used for growing strains of your bacteria. Generally, an experimental procedure will tell you what type of agar to use. If you are still unsure, okay, you can ask ang mga lab technician, kami, or kung ano man nga agar ang applicable sa type of microorganism ang ipagota. So, your scientist for microorganism. Not all types of your agar are good for students and to use unsupervised at home. The following list will give you an overview of a few types of your agar commonly found in the laboratory. Many of these can be brought from sa sa mga medical supplies. Okay? Now, you have your types of agar, brief description nila, and if it is suitable para sa mga students. So, you have your blood agar. It contains blood cells from animals, for example, sheep. Most bacteria will grow on this medium nga aman because the nutrients from the blood, sa aton nga sheep. Share ko lang as a medtech, so we use blood agar, but hindi kami ka-secure sang sheep blood. Therefore, we use human blood, amon man nga dugo. So, we take around 5 ml to 10 ml sang blood namon, then i-mix namon sa agar. Okay? Now, due to your potential of contamination from human contact. Now, you have here your chocolate agar, okay? Comprised by a sheep blood that provides the X and V factor, okay, necessary for a Haemophilus growth. This is a nutrient medium which is used in culturing your fastidious organisms such as your Haemophilus species and also your Neisseria. A chocolate agar, however, does not reveal hemolysis data. So, species differentiation among the members of your Haemophilus must be performed in another matter. So, if is this for the students man? Hindi man because the same lang gihapon due to potential contamination from human contact. Now you have here your LB or Loria bertagani agar. So, a subtype of a nutrient agar that is generally a medium for your microbiology student studies and may be used for routine cultivation of not particularly fastidious microorganisms. Also, does not preferentially grow one kind of bacteria over another. Okay, so we have a term here, your fastidious, okay, fastidious microorganism. So what are fastidious microorganisms? Fastidious microorganisms are those microorganisms that has a very complex nga requirement for them to grow. That's why ang mga like this one, like your LB agar or sa like your nutrient agar, okay, may not grow those type of microorganisms. So, it is not advisable so para sa mga fastidious na microorganism. Then you have your McConkey agar. So, this is an agar upon which only your gram-negative, okay, ano kayong gram-negative ka? They are, di ba, color red nga mga bacteria and can grow what is more likely your E. coli and will go into your red colony as they are pH indicator present. And it should be mentioned that your Makonki agar powder comes into two versions, one with a sugar lactose in it and one without any added sugar. Since your E. coli ferments sugar to acid, that's the red color. And one can add many different kinds of your sugar to this sugar-free nga Makonki nga agar and see if the red colonies are developed. If you get the red colonies, you know that it is your E. coli strain and you can use that agar or that your E. coli are using your sugar. And it is not suitable for student because due to selectivity. Selectivity ibig sabihin, usually for gram-negative or E. coli. And also for your E. coli, it is pathologic. Then you have your Miller's LB agar. Okay, so Miller's LB, you have your LB here. So it is Gloria Burgentini, pero gin-modified ni Miller. So this is common variation of your LB agar appears to have some components as the same as your LB, just in different portions. So yes, it can be used as mga students, but sticking with the generic formula as recommended. Then you have your Neomycin Agar. It contains antibiotic neomycin which can be found in many medications such as your creams, mga ointment, and also eye drops. Your neomycin discovered by the year 1949. Ang aton nga discoverer is your microbiologist Salman Waxman, and it produces naturally by your bacterium Streptomyces fasciidae. Okay, moreover, neomycin has a broad spectrum of effect. Ibig sabihin, it can kill both your gram-positive and gram-negative bacteria. It is relatively toxic to human and some people have allergic reaction to it. Often, your neomycin agar is used for the growth or culturing sa mga organism anaerobically. Your neomycin stops the growth of gram-negative bacilli and also your staphylococci, allowing your streptococcus nga mga species to grow abundantly. So it is not recommended sa mga student because due to safety concerns ko, some humans or it is harmful to people. Then you have your non-nutrient agar. They're usually not susceptible for bacterial growth, however, may be used for growing other microorganisms. Then you have your Na agar, okay, your Na agar, Na, okay, or nutrient agar. So, it will grow the largest number of different types of your microbes from your fungi and also your bacteria. Yet, you know, bacteria can grow on this. Some find it too rich and others find it too deficient. The nutrients in this beef broth and also some extracts from your yeast. Then you have your Sabarod's agar, used for fungi and has a low pH that will kill most bacteria. It also contains your gentamicin, which is aminoglycoside antibiotic. Your gentamicin can also treat many different types of your bacterial infections, particularly gram-negative infections. So same gihapon, no, because due to safety concerns. Then you have your TMA or your Thayer Martin agar. So it is a chocolate agar designed to isolate your Neisseria gonorrhea, known as your gonococcus, which is a species of a gram-negative bacterium responsible for the disease gonorrhea. So at least trivia lang sa aton, your gonorrhea gani class, amo na ang ga-cause or in layman's term it is your tulo. You know, due to your potential for contamination from human contact. Then you have your TSA. Your TSA stands for tryptic soy agar. Okay, is a basic medium used for culturing many kinds of your microorganism. Your TSA is mainly used in many initial growth medium for the purpose of observing your colony's morphology. Developing a pure culture, achieving sufficient growth for further biochemical testing, and also culture storage. Then it can be used for mga students. Then you have your XLD agar. XLD for your xylose, lysine, deoxy chocolate agar. So it is used for the culture of your stool sample. Your stool sample is sabihin tae. Okay, and contains two indicators. Is formulated to inhibit your gram-positive bacteria while the growth of your gram-negative bacilli is encouraged. Okay, ibig sabihin, inhibit, stop, yeah, or hindi siya magpag-grow sang gram-positive. Ang iya nga pagtubuon lang is your gram-negative bacteria. The colonies of your lactose fermenters appears to be yellow. No due to selectivity. Kay ang gram-negative lang ang pwede ka tubo. Onto our nutrient and broth and also sa aton nga agar plate, these are the most common growth media, although specialized media are sometimes required for your microorganism and also cell culture nga growth. Some microorganisms or organism term your fastidious, so kaya ginambal ko gina, okay, organisms needs a specialized environment due to their complex nutritional requirement. Okay, your viruses, for example, are obligate intracellular parasite and require a growth medium containing a living cell. That's why ang very analogous sa atong nga virus is kapi na sa mga pathogenic nga virus is they need a host to live. Pero what they do to their host is giving them illness kag masakit and also will reach to death na. So many human or microbial pathogens also require the use of human cells and cell lysates to grow on a media. Most common growth media nutrient broth or your liquid nutrient medium or ang imo nga LB medium or your lysogen broth are liquid. These are often mixed with an agar and poured into a petri dish to solidify. These agar plates provide a solid medium on which the microbe can be cultured. Okay? They remain solid as very few bacteria are able to decompose your agar. Many microbes can also be grown in a liquid culture comprised of a liquid nutrient media without your agar. So you have here an agar plate. So it is a solid gelatinous and there you can see growth or mga colony sang imo nga mga bacteria. We have here microbial pathogen growing on a blood agar plate. Okay, red blood cells are used to make an agar plate. Different pathogens that can use your red blood cells to grow are shown on this plate. On your left, okay, the left side is your Staphylococcus and on your right is your Streptococcus. Now, you have your definitive and undefinitive, defined and versus your undefined media. Okay, this is very important distinction between your growth media types. A defined medium will have a known quantity of all ingredients. For your microorganism, it provides a trace element and also vitamins required by your microbe and especially a defined carbon and nitrogen source. And your glucose glycerol or your glycerol are often used as carbon source. And ammonium salt or your nitrates as your inorganic nitrogen sources. Ang imo naman nga undefined medium has some complex ingredients such as your yeast extract which consists of a mixture of many chemical species is a known portion. And defined mga media are sometimes chosen based on a price and sometimes by necessity. Some microorganisms have never been cultured on a defined media. There are many differential types of your media that can use to grow your specific microbes and even promote a certain circular processes, such as wort, the medium which the growth of your media for the yeast that makes your beer. Without wort, in certain condition, your fermentation cannot occur and the beer will not contain alcohol or be carbonated or ang pagproduce ang bubbles. Now, your common or broadly defined nga culture media, you have your nutrient nga media. A source of your amino acid and also your nitrogen, for example, from your beef yeast extract. This is an undefined medium because the amino acid source contains a variety of your compound with the exact composition being unknown. This media contain all of the elements that most bacteria need to grow and also they are non-selective. So they are used for the general cultivation and maintenance of a bacteria kept in a laboratory culture collections. You have your minimal media that contains the minimum nutrient possible for a colony to grow, generally without the presence of your amino acids, and they are often used for microbiologists and genetics to grow a wild-type microorganism. This media can also be used to select for or against the growth of a specific microbe. Usually, a fair amount of information must be known about your microbe to determine its minimal media requirement. You have your selective media used to grow of only the selective nga microorganism. For example, if a microorganism is resistant to a certain antibiotic such as your ampicillin or your tetracycline, then the antibiotic that can be added to the medium in order to prevent other cells or mga microorganisms which do not possess the resistance from growing. And your differential nga media, also known as your indicator nga media, are used to distinguish one microorganism type from one another nga or one other growing microorganism on the same media. This type of media uses a biochemical characteristics of your microorganism growing in the presence of a specific nutrients or indicator, such as your neutral red, your phenol red, eucin Y, and your methylene blue. Added to the medium to visibly indicate the defining characteristic of your microorganism. This type of your media is used for the detection and identification of your microorganism. These are the few examples of your general types media providing the indication only. There are only your myriad of different types of media that can be used to grow and control your microorganism or your microbes. Okay, ato nga key points during this chapter is your culture media contains the nutrient needed to sustain a microbe or your microorganism. Your culture media can vary in many ingredients, allowing the media to select for or against your microbes. Your glucose or your glycerol are often used as your carbon source and your ammonium salt or your nitrates as inorganic nitrogen sources for your cultured media. And for the terms naman yung class, ang aton nga term for your culture is the process of growing your bacteria or other biological entity in an artificial medium or aton nga ginatawag ng mga agar or broth. Your lysogeny broth or your LB, okay, is a nutritional rich medium primarily used for the growth of bacteria. Now, for your complex and synthetic na media, okay, your key points here are defined media in made for the constituents that are completely understood. Undefined nga media has some parts of which is still not entirely defined or unknown. The presence of the extracts from the animals or other microbes makes a media undefined as your entire chemical composition of your extracts are not completely known. For the key terms, you have your recombinant. With this term refers to the composition. The term refers to something formed by combining existing element in a new combination. Thus, the phrase of recombinant DNA refers to the organism created in a lab by adding DNA of from another species or gina-modify mo. Okay, your serum niya class, okay, is the clear yellowish fluid obtained upon separating a whole blood into a solid and liquid component after it is allowed to clot. Also called as your blood serum. So, usually sa medtech na ang term. Okay, there are many type of your culture media in which your food that microbes can live on. Two major subtype of your media are complex and your synthetic medias known as your undefined and defined nga media. They have here undefined media. Luria broth, as shown here, is made up of a yeast extract. As yeast extract is not completely chemically defined, your Luria broth is therefore a undefined nga media. Undefined medium, okay, liwat as a complex ingredient such as your yeast extract, mga casein hydrolysate, which consists of a mixture of many chemicals, species in a normal portion. Undefined media are sometimes chosen based in the price and sometimes by necessity. Ang continuation sa kay nasa statement sa babaw. Some microorganisms have never been cultured or defined media. Defined medium, also known as your chemically defined medium or synthetic na nga medium, is a medium in which all of the chemicals used are known. No yeast, animal, or plant tissues is present. A chemically defined medium is a growth medium suitable for the culture of your microorganism or animal cells, including your human, of which all of the chemical components are known. The term chemically defined medium was defined by James and Smith as basal formulation, which may also be protein-free and comprised solely for biochemically defined low molecular weight constituents. A chemically defined medium is entirely free of animal derived components, including the microbial derived components such as your yeast extracts, and represents the purest and the most constituent cell culture environment. By definition, a chemically defined media cannot contain either fetal bovine serum or a bovine serum albumin or human serum albumin, as these products are derived from a bovine or a human source and contains a complex mixture of albumins and your lipids. The term chemically defined media is often misused in a literature to refer the serum albumin containing media. Ang animal serum niya class or albumin is routinely added to a culture media as a source for the nutrients and other ill-defined factors. Despite technically disadvantage to its inclusion and its high cost, technically disadvantages using your serum includes the undefined, okay, unknown, ano yung unod, nature of your serum, batch to batch variety in composition, and the risk of contamination. There are increased concerns about animal suffering inflicting the during your serum collection and adding the ethical imperative to move away from the use of serum whenever possible. Your chemically defined media differ from your serum-free tiga-tiga ba? Media in the bovine serum albumin or your human serum albumin with either your chemically defined recombinant version which lacks your albumin associated lipids or a synthetic chemical such as your polymer polyvinyl alcohol which can reproduce some of the functions of your serum. So let us proceed sa aton nga selective and your differential nga medias. Okay, you have here a rectal specimen for testing for your Neisseria gonorrhea. Liwat ang Neisseria gonorrhea in layman's term, it causes your tulo. Okay? And you have here a growth medium. We have an overgrow and in Neomycin Neisseria only wala sang grow. So you have a non-selective versus a selective media. This non-selective medium on your right, okay, on your right allows the growth of several differential bacteria species and is overgrown with your bacteria, your whitish lines, with the plates of while the plates on the right is a selective, ibig sabihin, Neisseria lang ang pwede ka tubo. Okay? Wala sang, ako may growing man na, so, because it's an image, okay, hindi ta ma-zoom gid. Check down. Okay? May mga white spots, giban. Where am I? Isa. Okay? Now, okay, yung may mga white spots, okay, that's the colony of your Neisseria. Ibig sabihin, they are the same nga sample. Okay? You have your chocolate medium. Okay, your chocolate medium is a non-selective. Therefore, any bacteria or microbes present in your specimen can grow. However, dili is a selective, your Neomycin medium, okay, only grows your Neisseria species. And you have here, my example man sa mga selective, you have Eosin methylene blue or EMB that contains methylene blue, toxic to the gram-positive bacteria, therefore it only grows your gram-negative. So, ibig sabihin, kung selective siya for your gram-negative, okay, patyo niya ang mga gram-positive. Term niya is it inhibits the growth of your gram-positive. You have your YM, okay, your yeast and mold, okay, which has a low pH, okay, deterring the bacterial growth. And you have your Neomycin Agar for gram-negative na mga bacteria. Your Hectoen enteric agar or your HE, which is a selective for your gram-negative bacteria. You have your mannitol salt agar, your MSA, which is a selective for gram-positive bacteria and differential for your mannitol. You have your terrific broth or your TB. It is used for a glycerol in cultivating your recombinant strains of your Escherichia coli. You have your XLB, xylose lysine deoxy chocolate, which is a selective for a gram-negative buffered charcoal yeast extract agar, and which is selective for a certain gram-negative bacteria, especially your Legionella pneumophila. You have your differential media or indicator media, distinguish one microorganism from one type or for one another growing on the same media. This type of media uses your biochemical characteristic of your microorganism, okay, growing in the presence of a specific nutrient or indicator, such as your phenol red. Though repeat lang isa do sa babaw nga introduction, so mas specific. You have your neutral red, your phenol red, eosin Y, methylene blue, added to the medium to visibly indicates the defining characteristic of your microorganism. This type of media is used for detecting or detection of your microorganism by a molecular biologist to detect the recombinant strains of your bacteria. You have mga examples here. You have your blood agar, used in strep test, which contains the bovine heart blood that becomes transparent in the presence of hemolytic. Okay? Then you have your Streptococcus eosin methylene blue or your AMB, which differentiates from a lactose and a sucrose fermentation. Your McConkey or MCK contains a differential for lactose fermentation mannitol salt agar for your MSA, which is a differential for mannitol fermentation. You have X-gal plates, which are differential for your lac operol mutants. Mga keywords ta sa among nga topic, you have your selective growth media. Selective, di ba? Only selects the growth of a desired organism and stopping the growth of altogether killing your mga non-desired nga organism. You have your differential. Media takes advantage of your biochemical properties of your target organism, often leading to a visible change when growth or if the target are met or present. You have your differential media, unlike your selective media. Okay, ang difference mo differential kag imo nga selective. Okay, your differential, okay, does not kill. Okay, it only indicates if a target organism is present. So you will have mga visible signs. And you have your recombinants, liwat, na define na nato meaning. You have your gene, a unit of heredity and a segment of your DNA or RNA that is transmitted from one generation to another. It carries your genetic information such as the sequence of amino acid or your protein. You have your anil, okay, one of the number of alternative form of a same gene occupying a given position or a chromosome. Now, your aseptic technique, dilution, streaking, kag spread sa plates. Kido nas ano to na ni sa previously sa kapina sa laboratory because it is very important to atunta to i-integrate again. You have your microbiologists rely on aseptic technique, dilution, on colony streaking and spread plate for day-to-day experiment. Key points ta. Aseptic technique is basically the mindset of keeping things free from contamination. As the word believe in has so many microbes that can interfere or makakontaminate sa atong mga experiments. Your colony streaking leads to the isolation of the individual colony, which are the group of your microbes that came from one single progenitor microbe. Then you have your spreading sa atong plate allow you to even spread the bacteria on a petri dish, allowing an isolation or mass separate nga colony growing for counting and further experiments. Now, key terms, you have your colony. A colony or bacterial colony is defined as a cluster of a bacteria growing on the surface of which they are solid medium, presumably cultured from a single cell. Your Bunsen burner, okay, a small laboratory gas burner whose air supply may be controlled with the adjustable hole. Then you have your microbiologists have many tools, but for relatively simple nga techniques are used in your microbiology daily. And then these are outlined here. Your aseptic technique or your sterile technique is used to avoid contamination of a sterile media and equipment during a cell culture. A sterile technique should always be employed when working with a live cell culture and reagent media that will be used for such culture. This technique involves using flame to kill contaminating microorganism and generally mode of operation that minimizes the exposure of your sterile media and equipment to your other contaminants. Okay, so you have here ang at tinatawag tapos nga serial dilution or to isolation pagit. Okay, your serial dilution is example of sa picture here you have an example of serial dilution of your bacteria in a 5-step. The diluted bacteria when the spread plate. Okay, or kung i-grow mo siya sa plate. You have here makuha ka sang 0.1 ml, butang mo siya sa 10 ml nga tube. Okay, nga mixed with H2O. Pero hindi ka magamit ng water from a tap, okay? You will be using a distilled water. A distilled water is a type of water or your mga NSS or normal saline solution ang ginagamit pang dextrose bila. Okay? Ang ginano is you put 9.9 ml then 0.1 ml from your broth, diba? That has bacteria on it. So it is 10 raised to negative 2. Then, i-culture mo na. Okay, you will see nga damo gyapon ang growth sa mga bacteria. Then, kuha ka sa first mo nga serial dilution, move mo sa isa. Okay, same gyapon yung you will be needing 0.1. Okay, mix with a distilled water nga 9.9 ml. Okay, as you can see, as gina-proceed mo ang imo serial dilution, mas gagamay ang colony growth sa ang imo nga mga bacteria. So, when working with the cultures of a living organism, it is extremely important to maintain the environment in which the cells are cultured and manipulated as free from other organisms as much as possible. This requires the exposure of your container or your sterile culture media to outside air should be minimized. Therefore, magamit ka dapat ara ka sa either hood. And the flame should be, and this requires the exposure of containers of sterile culture media uh, minimize. And the flame is used to resterilize the container, lids, and also sa iyang rim. This means passing rims and lids through the flame produced by a Bunsen burner. So doon nagpakita ko naman to sa aton nga video previously kung paano aton nga procedure sa uh, Asceptic Technique. Okay, in order to kill microorganisms coming in contact with those surface. Your sterile technique in general is a state of the being or mantra where every utilization of a sterile material comes with a caveat of taking every precautions to ensure it remain as free of your contaminants as much as possible for as much as possible. Heat is an excellent means of your killing your microorganism, and the Bunsen burner is the sterile technique's best friend. As, uh, Serial dilution is stepwise dilution of a substance in a solution. Usually, the dilution factor of each step is constant, resulting to a geometric progression of your concentration in a logarithmic function. A tenfold serial dilution could be 1 molarity, 0.1 molarity, 0.01 molarity, or 0.001 molarity, so on and so forth. Serial dilutions are used for accurately create highly diluted solution as well. A culture of your microbes can be diluted in the same function or fashion. For a tenfold dilution of a 1 milliliter scale, vials are filled with a 90 microliter or your microliter of water or your media, and a 100 microliter of a stock of your microbial nga solution are essentially being transferred. With thorough mixing after every dilution step, the dilution of your microbes is very important. To get to the microbes diluted enough to count on a spread plate. Here. Okay, so let us now proceed na sa aton nga stilking sa aton plate. So you have your 4 stilking plates here. Okay. So sa babaw, you have here the thicker lines. Okay, that is your first trick or halin gidya sa imong broth. Okay, or sa imong kuan sang specimen. You have your first trick, second, and third, and fourth, and you have your zigzag motion. So you can see the difference between your growth sa aton nga mga colonies from your streaking method here and sa zigzag method. Okay, maskin zigzag method siya, you can also see isolation sang bacteria. Okay, pero ang ilang pag-spreading is not as much mas presentable mas damo nga isolated ang diri sa stilking method sa may left side. So in the microbiology, the stilking is a technique that used to isolate a pure strain of a single species of your microorganism, often your bacteria. Samples can then be taken from resulting colonies of your microbial culture and can be grown on a new plate so that your microorganism can be identified, studied, or be tested. The streaking is done using your sterile tool or your inoculating loop, okay, such as your cotton swab, pwede mo na magamit, and also commonly your inoculating loop. It is deep in your inoculum, such as your broth or your patient specimen containing many species of your bacteria. The sample is being spread across your quadrant of your respective dish containing the growth medium, usually an agar plate such has been sterilized in an autoclave. Choice of which growth medium is used depending on your microorganism nga gina-target or being cultured or selected on amo nang gina-target. Okay, growth media are usually forms of your agar, a gelatinous substance derived from seaweeds. Okay, your spread plates or your agar plates are simply microbes spread in a media plate. Your microbes are in a solution and can be diluted. They are transferred to a petri dish with a media specific for the growth of the microbe of your interest or gina pangita mo nga microorganism. The solution is then spread uniformly through the number of possible means. The most popular is the use of your sterile glass beads that are shock upon the top of your media, spreading the microbes containing liquid heavily on the plate. Also common is the use of your bent glass rod, often referred as a hockey stick due to its similar shape. This glass rod is sterilized and used to spread your microbes containing liquid uniformly to your plate. Now, sa ato na nga special culture techniques. Your microbiologist would prefer to use well-defined media to grow your microbes, making your microbes easier to control. However, microbes are incredibly varied in what they use as their food source. Dahil kag isa pa ang ilang environmental requirement and the danger levels they have for humans and other organisms that they may compete with, or ang ilang pathologic kag isa pa is kung may mga uh, kung may damo pa mga wala pa na sa isolate, okay, so damo siya kaagaw sa nutrients. So therefore, the needs for a special nutrient or your growth environment is necessary to grow these difficult microbes. Your microbiologists often turn to the unidentified media, which is chosen based on the price and more so in this case by necessity. As some microorganisms have never been cultured on defined media, some culture media's conditions are relatively simple as demonstrated as a microaerophile. Ano ng microaerophile? Okay, is a microorganism that requires oxygen to survive but requires environment containing lower level of your oxygens that are present in your atmosphere around 20% concentration. More microphiles are also capnophiles. Okay, ano naman capnophiles? They require an elevated concentration of your carbon dioxide. Okay, capnophile requires higher or elevated concentration of your carbon dioxide. In the laboratory, they can easily be cultivated in your candle jar. Ano ko? May picture dito sa candle jar. Yeah. So, this is your candle jar. Ito simple na nasa nga jar. Okay. Ang purpose na is you put your your blood agar plate. Then, pasigahan mo sa sang candle. Kita nyo, ginilit ya. Then, i-close mo lang na ang imo nga jar. The main purpose of that is, di ba, ano kayo kinahanglan for combustion sa atong nga flame? Oxygen. Okay, so, ang oxygen diri sa jar will be consumed or will be burnt out sa atong nga candle and leaving your environment a carbon dioxide nga environment diin mas prefer sang aton nga microorganism. So that is the purpose of your candle jar. So a candle jar is a container which leads the candle, introduce sealing of your container and lead a candle. Okay, na-explain ko na. Liwat na. Oxygen per atmosphere in your jar. Many labs also has the access directly to your carbon dioxide and can add desired carbon dioxide level directly to the incubators where they can grow o sa aton nga macro aerophiles. Okay, ang ganamin niya ni sa mga lab nga may access to carbon dioxide is ang ila mismo nga incubator is a very modern, okay? Pwede ka naka-directly add carbon dioxide or pwede mo adjust ang environment sa imo nga incubator pa lang. So, therefore, dapat ano lang na imo ipagrow ang mastida because it can also kill other microorganism na ginapag-grow mo or inhibit ya sa imo nga incubator. So, animals and often the can be used to culture your microbes. For example, your armadillos are often used in the study of leprosy. They are particularly susceptible due to their unusually low body temperature, which is hospitable to leprosy bacterium, Mycobacterium leprae. The leprosy bacterium is very difficult to culture, and your armadillos have the body temperature of 34 degrees Celsius, similar to a human skin. Likewise, human can also acquire leprosy infection from your armadillos by handling them or consuming armadillo meat. Additionally, your syphilis, which is caused by the bacteria Treponema pallidum, is difficult to grow on a defined media. So rabbit are used to culture your Treponema pallidum. Treponema pallidum belongs to the Spirochaetophyllum or mga spirochaetes nga bacteria. Okay, to date, your spirochetes are very difficult, if not impossible, to rear in a controlled laboratory environment. This is also include other human pathogens, like your bacterium that causes your Lyme diseases. Okay, using your animals to culture human pathogens has problems. First, the use of the animal is always difficult for technical and ethical nga reason. Also, a microbe growing on an animal over than a human may behave very different from how the same microbe will behave on your human or ang iyang uh, effects. Okay, aside pa dabas may chance nga mag evolve muna. Then you have your some human pathogens are grown directly on the cells cultured from a humans, amplified by your bacteria Chlamydia trachomatis. The bacteria responsible for sexually transmitted diseases or infection or your STI in human known as your chlamydia. A chlamydia trachomatis only grows in human. The human cell culture known as McCoy cell culture is used to culture this bacteria. So you have here chlamydia's bacteria group and light microscopy view upon a cell infected with a chlamydia shown by the brown inclusion bodies. You have here the brown inclusion bodies. So these are the cells. So these are not uninfected. This is the infected. Then you have a large concern for your microbiology is trying to find a way in which human can avoid or get rid of your microbial infection. Typified by some of your above example, some microbes have to be grown in the lab, and some of them can infect humans. To deal with this, microbiologists use a classification of biosafety levels. A biosafety level is a level of your biocontainment precaution required to isolate dangerous biological agent in an enclosed facility. The level of the containment ranges from the lowest biosafety level, which is your 1 or your BLS1, to the highest at the level 4 or BSL4. The United States and also the Center for your Disease Control or your CDC may have specified these levels. For your 1, this level is suitable for work involving well-characterized agent not known to consistently cause disease in a healthy adult human with a minimal potential hazard to the laboratory personnel and aton nga environment. For your level 2, this is level similar to your 1 and it is suitable for work involving your agent of moderate potential hazard to personnel and the environment. It includes virus, bacteria, and viruses that cause only mild diseases or mild disease to human and are difficult to contact via aerosol in your lab settings such as your chlamydia because it is sexually transmitted disease. Then you have your level 3. Okay, this level applicable to clinic, diagnostic, teaching, research, and also production facilities in work is done with indigenous or exotic agents that may cause serious or potential lethal disease after inhalation or through aerosol. It includes various bacteria, parasites, and also your viruses that can cause severe to fatal disease in human, but for which treatment exists man. For example, your yellow fever. Okay, ibig sabihin, maski maka-infect pero at least it is treatable. Then for your biosafety level 4, okay, This level is reserved for work with dangerous and exotic agents that pose high-level individual risk of aerosol-transmitted laboratory infections, agents that cause severe to fatal disease in humans for which vaccines or other treatment are not available, such as Cerbolivan, Argentine hemorrhagic fever, Marburg virus, and also your Ebola. And then, ang subong nga, corona nga virus. Very few laboratories are biosafety level 4. So I have here a suit. Okay, it is a positive pressure suit. A scientist puts on a positive pressure suit. Something need to work with the most dangerous human pathogen in your biosafety level 4 to ensure nga wala sang may makasulod siya kag ma-inhale and ma-infect siya. You have your key points. Okay, microbes often those that we know little about have to be cultured with undefined media or growth conditions. The use of your animals to culture animals, okay, the use of animals to culture uh, is sometimes necessary and no simple media can be used. This presents technical and your ethical issue. And as human pathogens are often studied by your microbiologists, special safety conditions known as your biosafety level are used to keep researchers free of infection from the pathogens they study. And for your yellow fever, it's acute febrile illness of tropical regions caused by a flavivovirus and spread by mosquitoes, characterized by a jaundice, black vomit, and the absence of urination. For your Lyme disease, okay, it's an infection by a bacterium of a genus Borrelia, which is transmitted by a tick. Symptoms includes a rash, yellow fever, a rash followed by a fever, mga joint pain, and headache. So, that concludes our chapter 4.2. So, we'll be seeing sa chapter 4.3 because your bacteriology is like the introduction na gid sa mga microorganism kag para masapos ang study sa aton nga microbiology. We will be much focusing sa aton bacteriology. So, stay tuned lang class.